Hello everyone, this is Dr. Sangram Patil again. In this part 2 video of life processes chapter in your biology book for CBSE year 10, we'll be dealing with the nutrition part. So nutrition deals with the energy mechanism and also building the material that is important inside the body for building of various structures, repair and maintenance. So consider a person who is either running, walking or cycling. Then they are using up the energy. But even when they are not doing any apparent activity, energy is still needed to maintain a state of order in our body. And we also need materials from outside of the body to grow, to develop, to synthesize the proteins and other substances that we need inside our body. So this source of energy and the material comes from the food. Now, how do living things get their food? We need food, and it's a requirement. Without food, we all will die. But we get this requirement fulfilled in different ways. So some organisms will need some simple food material, and that will be obtained from inorganic sources in the form of water and carbon dioxide, and they will use sunlight to make energy from these inorganic sources. These organisms are called as autotrophic organisms or autotrophs. So these examples of these are plants, and some bacteria can make their food in this way. Whereas other organisms called as heterotrophs, such as animals and some fungi, they use complex substances. These complex substances are required to be broken down into simple, smaller substances, and then they can be used by the body to as a energy or for the growth purpose. To achieve this, the organisms use the catalysts. They are called enzymes. So they are biocatalysts inside the organism. The heterotrophs then use these enzymes to run their chemical reactions that extract energy and various food products from the raw material. So the survival of heterotrophs depends directly and indirectly on autotrophs. So as I said, the examples of these are the animals and the fungi. Okay, so autotrophic nutrition is very interesting. Let's go a little bit into details of autotrophic nutrition. So basically this is a photosynthesis mechanism that takes place in autotrophic organisms. It is the process through which autotrophs take the substances in from outside of the body and convert them into stored form of energy. So this material is taken in the form of carbon dioxide and water, as I have already mentioned, and in presence of sunlight and chlorophyll, these are converted into carbohydrates. So the carbohydrates are utilized for providing energy to the plant, and also carbohydrates that are not used immediately are stored in the form of starch, and this serves as the internal energy reserve to be used when the plant needs them in future. So in human beings and similar organisms, such a stored energy is not in the form of starch, but it is in the form of glycogen. So excess glucose, carbohydrates, they get stored as glycogen in human body. So now let us look at this equation of photosynthesis and the few details of process of photosynthesis. The carbon dioxide and water in presence of chlorophyll and sunlight, they will be converted into glucose, oxygen, and there will be also a water on this side. So to balance this equation, so glucose will be C6H12O6. Okay, to balance this equation, we have to put 6 here, 12 water, and again 6 oxygen and 6 water molecules. So 6 CO2, 12 H2O in presence of chlorophyll and sunlight will give rise to 1 molecule of glucose, that is C6H12O6, 6 molecules of oxygen and 6 molecules of water. Now let's see what are the steps involved in photosynthesis. So these are the 3 main steps of photosynthesis. The first, and these steps may not take place exactly one after the other. They may take place with some gap between them. So first step is light absorption by chlorophyll. Second step is conversion of this light energy into chemical energy, and at the same time the water molecule splits into hydrogen and oxygen. The third step is carbon dioxide is reduced to carbohydrates. So this is a reduction reaction. This is reduced. Okay, so remember this. Now let's have a look at the diagram for this mechanism. So here is a diagram for the structure of the leaf that is involved in photosynthesis, and let's go through this very briefly because you're not required to know it in great details at this stage. But these steps take place in relation to the leaf, and we have the lamina or the leaf blade here, and you can see the midrib that goes through the center of this leaf, and there are veins that are given up from the central midrib, and this is the cross section of this leaf they have taken and given in great details. So this phloem and xylem vascular bundle system is shown here in this diagram, that includes the waxy layer of cuticle. Then underneath that is upper epidermis, so that's a cuticle layer, and then you have epidermis, that's a single layer of flat cells. These are the external covers, the outside layers of this leaf, and underneath that this is a spacious compartment that contains chloroplasts. So the cells that have got chloroplasts, and there are lots of air spaces around these cells, and at the other end of the leaf, on the other surface of the leaf, is the lower epidermis. So cuticle, upper epidermis, lower epidermis, and the lower epidermis has got these pores which are guarded by the guard cells. And as you might remember from your previous year's biology classes, these guard cells, they actually control the size of the pores. So when there is enough water in them, they swell up, they are turgid, and they open up the pore. When they lose water, when they dehydrate, they shrink and then they close the pore. So they maintain the aperture size of this pore, and then depending on that, the gaseous exchange, carbon dioxide and oxygen, as well as the vapor, water vaporizes through these. So these processes can be controlled with these guard cells and pore mechanism. So now the 3 steps I mentioned, the 3 steps of the photosynthesis, that is light absorption, then light conversion into chemical, and water breakdown into hydrogen and oxygen, and reduction of carbon dioxide. These may not take place one after the other immediately. So for example, in desert plants, they take up carbon dioxide at night, and then they prepare an intermediate substance that will be acted upon by the energy of the sunlight absorbed by chlorophyll, and this process will take place during the day. So CO2 absorption takes place during night, and energy action after light absorption will take place during the day. So now let's see how each component of this reaction is necessary for photosynthesis. So let's that is the thing we have seen in the cross section of this leaf, the diagram I shown you. So these cells have these greenish dots, they are chloroplasts, and these green dots, these are the plant cell organelles, the intracellular organelles. They contain chlorophyll, and the chlorophyll is important, the most important component of the process of photosynthesis. Now your book, the CBSE book, gives you an experiment, and this experiment wants you to perform a starch iodine test. So here is a starch iodine test. So you can see different substances and elements required for this test. This is a spirit lamp, and there are 4 containers here, and we'll be needing 1 test tube as well. So this petri dish has got a green leaf, and there is a water in this beaker, and is an iodine solution, and this is an alcohol solution. So first step is to start boiling this water and put the leaf into this water. So that's the first step here. So this will make the leaf soft, so that you can put iodine drops on the leaf, and they can be then absorbed easily by the substances in the leaf. Now once it's become soft, you put that leaf into the alcohol, into the test tube, and add some alcohol to that test tube. Now put that test tube into the water that is being heated. So you will have more loosening up of the tissues of the leaf there. Now once this is done, you take that leaf out and put it into the petri dish and pour few drops of iodine on that leaf. Now remember, this leaf tissue has become very soft, so this iodine will get absorbed easily into this leaf, and the leaf will turn something like this. So now, why has this turned bluish? The reason for that is this iodine is absorbed by the starch in this leaf, and then iodine turns that leaf, the color of that leaf, changes the color of that leaf. Now your book gives a slightly different experiment. It wants you to take a potted plant with variegated leaves like money plant, or crotons, and then they ask you to keep that plant in the dark room for 3 days. So there will be no photosynthesis during these 3 days. And the starch from the leaf will be used up, consumed partially during these 3 days. So when you actually look at that leaf, you will see that leaf has changed its color because the starch will be used up. So green areas will be lacking in certain parts. So they ask you to plot the size of this onto the paper, and then they ask you to perform these types of starch test, like boiling, putting the leaf into boiling water, then into alcohol, heating it up, and then adding iodine onto it. So at the end of the experiment, you will get something like this. The starch, leftover starch in the leaf, will be converted by iodine dye into this type of color, and that will correspond with the pre-test kind of distribution of green color. So this shows that iodine is getting absorbed on the surface of the starch molecule, starch stores there, and then it changes the color. So this is the starch iodine experiment your CBSE book has mentioned. Now we'll go to the next topic, that is, how do plants obtain carbon dioxide? Now that's a very interesting topic again. So as I have mentioned to you, that the plants get carbon dioxide from outside, and we have seen this last year. We have seen this these diagrams of the leaf surface, and they have a stomata on the surface that surround the pore, and there are guard cells. Guard cells will be in two states: either they will be swollen, turbid, that will open up the stomatal pores, or they will be shrunk and closing the stomatal pore and stopping the exchange of gases as well as water vapor. So exchange of gases occurs across the surface of the leaves, roots, and stems as well. And so there is a large amount of water that can be lost through the stomata. The plant closes these pores when it does not need carbon dioxide for photosynthesis. The opening and the closing of the pores is a function of guard cell, and guard cell will, depending on whether it's swollen or shrunk, it will cause the the gate opening or closing mechanism in action, stay in action. So next, your book talks about an activity for you, and this activity is important. I'll take you through this activity. So they ask you to take two healthy potted plants of the same size, keep them in a dark room for 3 days, so there will be no photosynthesis during this period. Now place each plant on a separate glass plate. Place a watch glass containing potassium hydroxide here by the side of the pot, but only in one pot, not in the other. And the potassium hydroxide, you might remember, it absorbs carbon dioxide. Cover both the plants with separate bale jar so that you can observe from outside. And they want us to seal the junction, the bottom of the jar, with some Vaseline, so there is no escape of oxygen or carbon dioxide from these bale jars. And keep the plant in sunlight for 2 hours, and pluck the leaf from each plant and check for the presence of starch, as we have done in the starch iodine test. So the question is, if you do this, which plant will show the starch, show up the starch, and which plant will not show up the starch. So now the explanation for this activity is that potassium hydroxide absorbs all the CO2 from this bell jar. So there's no CO2 available. And remember your photosynthesis definition, it is the production of energy using carbon dioxide, water, chlorophyll, and sunlight. So if there's no CO2, this plant will not be able to perform photosynthesis. So there will be no starch in this lip because this plant has not done photosynthesis for last 3 days. It was kept in the dark, and now you are taking the CO2 away. Now this plant has received sunlight, it has a CO2, so it will produce photosynthesis and it will show the presence of starch. So if you do iodine test on this, the color of this leaf, the leaf from this plant will change to blue, and this will not change. So that's the second activity your book gives. And there's one more activity they talk about. We'll also talk about that in next few minutes. But before that, let's finish up the autotrophic nutrition topic with these few notes. So autotrophs also need raw material for building of their structure, maintenance, and repair, and all that kind of activities. So it's not just water, carbon dioxide, chlorophyll, and sunlight, but they need other substances as well. So water comes from soil. They also need nitrogen, phosphorus, iron, manganese from soil. And nitrogen is the substance that basically they can also take it from bacteria. These bacteria fix the nitrogen from environment, and it's called nitrogen fixation, or it can come from inorganic nitrates and nitrites. So nitrogen is used for proteins, for making proteins and other compounds. So in the end, the very important topic, heterotrophic nutrition, and this will include the organisms, including the human being. Now each organism is adapted to its surrounding environment, and the nutrition it takes will depend on the food material that is available, its type, and as well as what the organism needs to do to obtain this nutrition. For example, if the food source is stationary like plants, grass, or it's mobile, like if some animal has to rely on other animals like deer, then that will allow different type of access to different animals, that will need different type of access to different animals to obtain their food. And also that will also make changes to the nutrition machinery, nutritive apparatus used by these animals. So the strategies that are used to take the food by the organism will differ. Some organisms will break the food down into smaller pieces outside the body and then take that food in the body. Example for these are fungi. So they break down food outside and then they take it inside. So fungi do that, like bread molds, yeast, and mushrooms. Other animals take the whole food material and then they break it down inside the body. Okay, so what can be taken in and broken down depends on the body design of that organism and how the body functions. Some other organisms, they use parasitic nutritive strategy. So that means they rely on plants or animals without actually killing them. So this parasitic nutritive strategy is used by various organisms. So let's discuss three types of heterotrophic nutrition strategies briefly. Okay, so you don't need to know them in great details, but let's have a brief introduction. So heterotrophic is you are dependent on plants to plants or other animals to survive for your nutrition. So you can be saprotrophic you can be relying on saprotrophic nutrition, parasitic nutrition, or it could be holozoic nutrition. So saprotrophic strategy is to rely on dead plants, dead and decaying animal bodies, example yeasts and mushrooms. Okay, so that's a saprotrophic nutrition. Then parasitic is you are feeding on another living organism like tapeworm or lice. Okay, and holozoic nutrition is ingestion of organic food material like we do. We eat organic food material, so human being or amoeba. So parasitic nutrition, which I just mentioned, is actually a mode of heterotrophic nutrition where a parasitic organism lives on the body surface or inside the body of the host and gets nutrition directly from the host, the body of the host. And various examples for this are cruscuta. In our Hindi and Marathi languages it's called amarbel, ticks, lice, leeches, tapeworms, mosquitoes. Yeah, so these are the parasitic. They don't kill the organism, but they get the food either from external or inner surface of the organism directly. So they are parasitic organisms. Okay, now how do the organisms obtain their nutrition? Okay, so let's go a little bit more detail into this. So unicellular organisms differ in the way they take nutrition from the external surface, and the digestive systems are different in different organisms based on how they take the food. Unicellular organism, as you can see in this diagram, their body surface is in contact with the external surrounding from all over, and all the body systems, they are in this single cell. So all the systems are in contact with the surrounding environment. So food is taken inside from the entire surface. It can go from any side in and enter into the cell. But as the cells become complex, the organism develops specialized systems to perform different functions. So amoeba, I have just explained you that the food is taken in using finger-like pseudopodia. They are extensions, and they are temporary finger-like extensions. They come out of the cell surface and then they fuse. They grab the food particle and the finger-like projections fuse with each other, and the food particle is then trapped inside a food vacuole. Okay, so that's a food vacuole, and then food vacuole just mixes, it merges with the rest of the cell, and the food particle then gets digested into simple substances. It's the complex substance is broken into simple substance, and the remaining undigested food is then excreted out from the cell surface itself. Okay, and the same is the process in paramecium, but Paramecium differs from amoeba. Amoeba can take food from any surf point. Paramecium has got a single oral groove on its surface, and the food particle is pushed by the cilia on the surface of the paramecium towards the oral groove, and then the food enters through the oral groove, and then it goes into the cytoplasm. It gives rise to food vacuoles. They are digested, and the rest undigested food is excreted from the cell membranes, from the cell surface. So that is how unicellular organisms, they carry out their nutrition. So we've seen two examples, amoeba and paramecium. Now let's look at the human nutrition, and that's very important for us. So for this, I'm going to discuss whatever is given in your CBSE book. So they first of all give you a test to do, the activity to do, and this activity, they have asked 1 ml of starch solution to be taken in 2 test tubes, A and B. Add 1 ml of saliva to each of them, and then the 1 ml of saliva just to the test tube A and nothing to test tube B. Leave it for half an hour, and then add few drops of iodine into each test tube. So one has a saliva and starch solution, the other has only starch solution, and then you add iodine drops, and they ask what will be the color of the solution in the test tube. So as you can see, the one with saliva and starch turns reddish, but without saliva and only starch, you remember from your previous experiments, activities, that starch turns bluish when you add iodine to the starch solution or starch containing leaf. So here, what is happening? Why there is starch here and there is no starch reaction here? So the answer lies in the fact that saliva has got the chemical substances that are biological catalyst, enzymes. They convert starch, they break down the starch, and there's no more starch left here. That's why you can see this is turned red, and where the starch is there, test tube B has turned blue because of the presence of the starch. So we eat various food types that pass through the digestive tract, and naturally the food has to proceed to generate small particles of similar size and texture. This is done by crushing the food with our teeth. Since the lining of the alimentary canal, that is our gastrointestinal tract from mouth to the anus, is soft, the food is also softened, is made weight to make it pass smoothly through the food pipe. When we eat, our mouth sometimes waters. This is not just a water, it's a saliva secreted from our salivary glands inside the mouth. The another aspect of the food we ingest, that it is a complex in chemical nature. If it is to be absorbed from the alimentary canal, it needs to be broken into the smaller molecules, and this is done with the help of enzymes. And saliva contains salivary amylase that breaks the starch down from complex molecule into simple sugar. The food is mixed thoroughly with saliva and moved around the mouth while chewing by the muscular action of the tongue. So the tongue helps to move the food around the mouth. The saliva is mixed in it, so the combined action makes it smooth. The salivary amylase starts acting on it to convert starch into the glucose. Now food has to move from mouth to the further lower structures of the digestive tube. So this movement in a regulated manner along the digestive tube is the property of this canal. The muscle in the wall of this canal that contracts rhythmically in order to push the food forward from mouth towards the lower structures of the alimentary canal. These rhythmic contractions of the muscles in the alimentary tract or gut canal, these movements are called as peristaltic movements, and they occur all along the gut. From mouth, the food enters into the esophagus. It is also called as food pipe. From esophagus, it enters the stomach. It is a large J-shaped bag, and muscular wall of the stomach here helps to mix the food thoroughly with more digestive juices. Remember, we saw saliva and its digestive enzymes in the mouth. Now stomach has got more digestive juices. These digestive functions in the stomach are taken care by gastric glands. They are present in the wall of the stomach. These glands, they release three important chemicals. The one is pepsin, the second is hydrochloric acid, and third is mucus. Okay, now from stomach, the food enters into the small intestine, and between stomach and small intestine there is a sphincter, that is a small constriction that controls emptying of the food from stomach into the small intestine. So this is the small intestine, and that's where the sphincter lies. There is a small stricture constriction that controls the passage of food from stomach into the small intestine. So the food digestion function in the stomach is important, as hydrochloric acid creates acidic medium for the action of the enzyme pepsin, and hydrochloric acid helps in this way because then pepsin can break down the protein in the stomach. So other functions are also carried out here. There is absorption of certain nutrients in that takes place inside the stomach, and also acid helps to neutralize various bacteria and microorganisms that may enter along with the food. So food, as I said, as it enters into the small intestine through the sphincter, the small intestine is the longest part of the alimentary canal. So this is a quick recap of what happens in the stomach. From esophagus, the food enters the stomach. It's a big muscular J-shaped bag, and through the sphincter, the food enters into the small intestine. So muscular contraction of stomach walls mechanically digest food. These muscular contractions are called peristalsis. Gastric juice that comes from gastric glands that are there in the wall of the stomach, that activate It contains hydrochloric acid that activates pepsinogen, that converts pepsinogen into pepsin, and pepsin is important for digestion of the protein. So pepsinogen converted into pepsin in presence of hydrochloric acid. Pepsin breaks down protein into small peptides and amino acids. So the pH here is quite low. Low pH means the environment here is acidic. So that is about the things that take place in stomach. Now, small intestine is the longest part of the gastrointestinal tract, and it's very important in digestion. So let us look into few details of what happens in small intestine. So length of the small intestine is different in different animals depending on what type of food they eat. Herbivores, they eat grass. Okay, so if the animals are herbivorous, then they will need more length of small intestine, so they have long small intestine. Whereas carnivores like tigers, they eat meat. The meat is easy to digest, so they need shorter small intestine. The small intestine receives different digestive juices, and the digestion of carbohydrates, proteins, and fats takes place in small intestine. So protein, carbohydrate, and fats, they are digested. So proteins are broken down into amino acids. Carbohydrate complex sugars are broken down into simple sugar like glucose, and fats are broken down into glycerol and fatty acids. So this is a very important function here, and for this to happen, the small intestine receives different juices for digestive function. It receives juices from liver, from pancreas. So if you look at this diagram, so juices from liver, juices from pancreas, and it also secretes its own juices. These are intestinal juices. Okay, we have seen that in oral cavity there is saliva secreted. In stomach there are gastric juices. In small intestine there are juices from liver, pancreas, and also from intestine. So we'll go a little bit detail into this. So bile from liver. Bile is a mixture of the secretion that comes from liver. It is stored in a gallbladder, and then it comes into the small intestine. It helps to break fat into small droplets for easy digestion. The pancreatic juice from pancreas, this contains carbohydrates, protease, and lipase. So you can see from the name, ASE, when they say ASE, that is something that breaks down carbohydrate. Protease breaks down protein. Lipase breaks down lipids. So these are enzymes from pancreatic juice. And then you have intestinal juice from small intestine. This also contains carbohydrates, protease, and lipase. So very important in digestion of all the three major food components, that is proteins, carbohydrates, and fats. So now let us talk a little bit further into this. So food coming from stomach is acidic, and so whatever comes from stomach is acidic, and it needs to be neutralized, because then acidic food will not be useful in the small intestine area. So it is made alkaline for the pancreatic enzymes to act. Bile juices that come from liver and stored in a gallbladder, they are secreted into the small intestine. These bile juices, they act on the fats. Fats are present in the intestine in the form of large globules, which make it difficult for enzymes to act on them. Bile salts, they break the fat down into smaller globules, so they increase the efficiency of enzyme action. This is uh, similar to the emulsifying action of the soap on the dirt that we have seen earlier in your other chapters of the science book. The pancreas secrete pancreatic juice, which contains enzymes like trypsin for the digestion of protein, lipase for breakdown of fat. The walls of the small intestine contain glands that secrete intestinal juice. The enzymes present in it, they convert protein into amino acid, complex carbohydrate into glucose, and they convert fat into fatty acid and glycerol. The digested food is then taken up by the walls of the intestine. Now have a look at the wall of the intestine. You don't need to know great details of this diagram, but I've just tried to show you how the wall looks microscopically. The inner lining of the small intestine has numerous finger-like projections, and these are called as villi. V-I-L-L-I, villi. Single is villus and multiple is plural is villi. They increase the surface area for absorption. Okay, so there's so much surface area available for absorption. That is why there's a small projections here. The villi are richly supplied by blood vessels, and they absorb the food that is taken into these blood vessels, and then that is distributed to the cells of the body for to supply them energy and the building material for maintenance, repair, and growth. The unabsorbed food from small intestine then goes further into the large intestine. So you can see this is a small intestine, the inner structure. It enters here into the large intestine. You can see this is a large intestine, okay, that ultimately goes to the anus, to the outside environment. So small intestine, stomach, esophagus, oral cavity, mouth, and from large intestine it enters, it opens at the anus. So large intestine also has got villi to absorb the food, especially the water from the leftover material, and the rest of the material is removed from body via anus. The exit of waste material is regulated by anal sphincter. So there is another sphincter here that has a voluntary control. So we control the exit of the waste material. This is similar to the exit of the waste water like urine. So you have sphincter at the base of the bladder and in relation to the urethra. Now we'll quickly look at the function of the teeth. As I have already said, the food in the mouth is broken down with the action of the teeth, and then with the muscular action of the tongue, it is mixed with this salivary secretion. So these teeth can sometimes develop dental caries, or it's also called as tooth decay, and it causes gradual softening of the enamel and dentine. So these are the tough membranes of the tooth. It begins when bacteria acting on sugars produce acids. Acids soften or demineralize the enamel of the tooth. So that enamel of the tooth is softened or demineralized by acid. And who produces acid? Bacteria in the sugar. Masses of bacterial cells together with food particle stick to the teeth. They get stuck in the small crevices in relation to teeth to form dental plaques, and saliva cannot reach the tooth surface to neutralize the acid, as these dental plaques, they cover the teeth. So saliva can neutralize the acid, but because the dental plaques they develop on this surface, because of the food particles getting stuck there, then saliva cannot neutralize the acid, and that acid softens the enamel, and then that develops into dental caries or tooth decay. If we brush the teeth after eating, it removes the food plaques before the bacteria can produce acid. And if we don't treat this, microorganisms can invade the pulp and they can enter inside, causing infection or inflammation. So this is about human nutrition. I hope you enjoyed this. In next video, I'll take you through the respiration as a life process, and I'll see you soon. I'll post that video soon on the channel here. Take care. Bye bye.